Hi, welcome to SGT University. I'm Dr. Swati Singh from Faculty of Allied Health Sciences. Today we will be studying about citric acid cycle.
Topics for this e-lecture are Introduction of citric acid cycle, Reactions of citric acid cycle, Energetics, and Regulation of citric acid cycle. Citric acid cycle is a series of reaction that produces two carbon dioxide molecules, one GTP, and reduced forms of NADH and FADH2. It is a series of enzymatic reactions that occurs in all aerobic organisms. It involves the oxidative metabolism of acetyl units and serve as the main source of cellular energy. It is also known as tricarboxylic acid cycle, as there are three carboxyl groups present on its first two intermediates, and also known as Krebs cycle after its discoverer's name, that is hence Krebs. In eukaryotes, the citric acid cycle takes place in the matrix of the mitochondria. In prokaryotes, these steps takes place in cytoplasm. The citric acid cycle is a closed loop, that is, the last part of the pathway reforms the molecules used in the first step. It includes 8 major steps. The breakdown of sugars by glycolysis yields pyruvate that in turn is decarboxylated by the pyruvate dehydrogenase complex, generating acetyl coenzyme A, which is the starting point for the citric acid cycle. Reactions in the citric acid cycle. The first step is a condensation step where the 2-carbon acetyl group combines with 4-carbon oxaloacetate molecule to form a 6-carbon molecule of citrate by the enzyme citrate synthase. In the second step, citrate is converted into its isomer isocitrate by the enzyme aconitase. In the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule alpha ketoglutarate. During this step, NAD is reduced to form NADH. The enzyme catalyzing this step, isocitrate dehydrogenase, is important in regulating the speed of the citric acid cycle. In the fourth step, alpha ketoglutarate is oxidized to form 4-carbon molecule. This binds to coenzyme A, forming succinyl coenzyme A. A second molecule of NAD is reduced to form NADH and produced a second molecule of carbon dioxide. The enzyme catalyzing this step is alpha keto dehydrogenase, which is also important in the regulation of the citric acid cycle. In the fifth step, succinyl coenzyme A then converted to succinate in the presence of enzyme succinyl coenzyme A synthetase, and one molecule of GTP is produced. Succinate is converted into fumarate by succinate dehydrogenase in the sixth step, and one molecule of FADH2 is produced. In the seventh step, fumarate is converted to malate in the presence of enzyme fumarase. In the last step, malate is then converted into oxaloacetate by malate dehydrogenase. The third molecule of NADH is also produced. Next comes the energetics of TCA cycle. Each cycle produces 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of GTP. GTP is energetically equivalent to ATP. Each molecule of glucose produces 2 molecules of pyruvate, which in turn produce 2 molecules of acetyl coenzyme A. Or each molecule of glucose produces 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of GTP. These carriers will connect with electron transport chain to produce further ATP molecules. Come the regulation of TCA cycle. The three rate limiting steps are catalyzed by citrate synthase, isocitrate dehydrogenase, ketoglutarate dehydrogenase. Acetyl coenzyme A, which is the starting material, is regulated by the controlled amounts of pyruvate that is converted into acetyl coenzyme A in the presence of pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex reaction is inhibited by ATP, NADH, and is activated by AMP, calcium, and NAD. Citrate synthase is responsible for the first step of the cycle when the acetyl coenzyme A is combined with oxaloacetic acid to form citrate. It is inhibited by high concentrations of ATP, acetyl coenzyme A, and NADH, which indicates an already high level of energy supply. Therefore, the rate of the reaction is reduced when the cell has high level of ATP. The molecule produced in the reaction citrate can also act as an inhibitor of the enzyme. ADP works as an allosteric activator for this enzyme. The enzyme isocitrate dehydrogenase is an important catalyst in the third step. It regulates the speed at which the citrate isomer isocitrate loses a carbon to form 5-carbon molecule, that is alpha ketoglutarate. The coenzyme NADH is a product of the reaction, and at its high levels it acts as an inhibitor by directly displacing NAD molecules it is formed from. Enzyme alpha ketoglutarate dehydrogenase is another important catalyst in the fourth step of the cycle, where alpha ketoglutarate also loses a carbon and combines with coenzyme A to form succinyl coenzyme A. The two products of the reaction, that is succinyl coenzyme A and NADH, both works as an inhibitors at large concentrations. The calcium ions also activate the enzyme isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase.
So, today we covered about citric acid cycle. In our next lecture, we will be studying about glycogen metabolism. Till then, keep learning, keep growing. See you next time.